Good morning, everyone, and thank you for joining us to the webinar of COVID-19 antigen-specific B and T cell research tools. In this webinar, I will present to you with relevant applications data and workflows. And at the end, I will be happy to answer your questions. The first question to ask is, what is the importance of antigen-specific T and B cells in SARS-CoV-2 pandemic? A decade after the MERS outbreak and 2 decades after the SARS epidemic, we do not have any approved preventive or therapeutic agent available. Studies have been shown that the neutralizing antibody titers and the memory B cell response are short-lived in SARS-recovered patients, and the antibody will target primarily the homologous strain. Recent publications have been revealed important insights into the immune response of patients who are hospitalized. Similar to other respiratory viral infections, the adaptive immune response, particularly of T cells, have a prominent role in the SARS-CoV-2 infection. However, it remains unclear whether the T cell responses are helpful or harmful in COVID-19, and whether T cell responses are suboptimal or dysfunctional or even excessive. Since evidence have been provided for both ends of the spectrum, therefore, the production of SARS-CoV-2 reactive T cells is an option as an adoptive cell therapy treatment. This webinar will be divided into 2 parts. One for the antigen-specific T cells, and the second one for the antigen-specific B cells. I will start with the T cell section. Let's have a close look on how we can define the antigen-specific T cells. Of course, all T cells are antigen-specific. However, we are interested to explore those T cells that were specifically activated by the coronavirus prior to the analysis. Hereby, only T cells of the given specificity are activated and clonally expanded. The initial specificity of those antigen-specific T cells is very, very low. The analysis of the activated antigen-specific T cells is very important for the basic understanding of the immune response and plays a pivotal role not only in the infectious diseases, but also in vaccine development, immunomonitoring, autoimmunity, and many more. A few words on the activation process of the T cells. Upon antigen-specific activation, T cells are clonally expanded, upregulated activation markers, and release cytokines. There are 2 types of stimulation. There is the polyclonal stimulation via the CD3, CD28 stimuli, or antigen-specific with the use of either peptides, peptide spools, proteins, and cell lysates in order to cover all T cells epitopes, and of course, in the presence of antigen-presenting cells. Most importantly is that all relevant information can be obtained using a flow cytometry device and selective markers for cell enrichment and isolation. In example, CD69 for CD8 T cells and CD154 for the CD4 T cells. When working with antigen-specific T cells, there is a challenge to overcome. Those cells are very rare and in extremely low frequencies, depending on the individual experience and the expression of the specific antigen itself. As you can see, the frequencies are between 0.1 to 0.001% of the whole T cell population. Different analysis methods need to be considered upfront, and phenotyping markers need to be chosen. Because at the end of this is like finding a needle in the haystack. After the short introduction, let's dive into the workflows. We will begin with the research tools and workflow. Afterwards, I will talk on the importance of those applications in the clinic. In research, we can divide the workflow into two sections. The analysis section, which includes the all-in-one kit in which I will present in a few minutes, and isolation, which includes two main workflows. Both sections are based on a product named the Peptivators from Miltenyi Biotech. So, what are those Peptivators? As presented in the figure on the right, the Peptivators are peptides consist of 15 mer length with 11 amino acid overlaps between them, covering the respective sequence of the antigen. They can be used to load antigen-presenting cells and to stimulate the SARS-CoV-2 specific T cells. Those peptides pools are causing highly and efficient stimulation of both CD4 and CD8 T cells. After the stimulation, T cells can then be detected or isolated for further research, including analysis of T cell immunity after vaccination, or even monitoring the immune status after infection. On the left side is the list of all the SARS-CoV-2 peptides pools that are available, including the UK and the South Africa variants. As you can see, for the UK and the South Africa variants, the spike or the end protein are becoming as mutated region and also with the wild type reference. In the next slide, I will show you the easy and fast workflow of this kit stimulation and analysis kit. For the stimulation and analysis application of the SARS-CoV-2 antigen-specific T cells, we offer the all-in-one kit. I wish to point out that each of the kit components can be purchased separately if you already have an established working protocol. However, if you only have the samples, then this kit is already calibrated for your system. So what does this kit include? It includes the SARS-CoV-2 Peptivators of your choice. You need to choose which antigen you wish to stimulate with. A positive control stimulation, which is the reagent called the cytostim. This is a polyclonal stimulation, antibody-based. A live dead marker, which called the viability dye on the violet laser. And 8 flow panel antibodies for the immunotyping of the cells. Fixation and permeabilization buffer set for the intracellular staining of the cytokine that are secreted based on the stimulation. And of course, the Brefeldin A, which is the Golgi inhibitor in order to accumulate the cytokine inside the cells. In the next slide, I'm going to show you the schematic overview of the workflow. This is a very easy and fast protocol. The first step is the sample collection and the human PBMC isolation. The second step is the activation using the Peptivators peptide pools. And after 2 hours, you adding the Brefeldin A for another 4 hours. The entire activation step is taking 6 hours. Afterwards, there is a staining with the live dead cell and the 8 antibody phenotyping panel. Then there is the fixation and permeabilization step and the intracellular staining for the cytokine secretion and detection. At the end, the samples are being analyzed by a flow cytometer that is available in your lab. In the next two slides, I'm going to present two analysis strategies that has been done with this kit. The presented plots are an exemplary data showing interferon gamma producing CD3 T cells. The data demonstrate an increased number of activated T cells in recovered CD19 patients upon stimulation with the SARS-CoV-2 peptide pools. A negative control, samples that were left untreated without any addition of the antigen. This is one way to analyze and present the results. As you can see, the plots are CD3 cells against the detection of the interferon gamma cytokine upon stimulation. In this slide, there is an analysis example of CD4 and CD8 T cells from PBMC samples of a reactive donor. The PBMCs were generated from a blood sample of SARS-CoV-2 reactive donors, stimulated or left untreated. A mix of CMV and EBV peptivators was used as the positive control, as you can see on the right. Subsequently, after pre-gating on CD4 or CD8 cells, respectively, the expression of the activation markers and cytokines, it is clearly shown that SARS-CoV-2 reactive T cells express activation markers and cytokines after the stimulation with the respective peptivators. The respective peptivators appears in the middle, as you can see, with protein N, protein M, and the S, which is the spike protein. These results were obtained with the recombinant engineered antibodies from Miltenyi Biotech, presented in the table on the right side of the slide. In the next slide, I'm going to present the two methods for the antigen-specific T cell isolation. These are two main workflows for the enrichment and isolation of those rare population. One is the cytokine secretion assay, a unique tool. This tool allows the detection and enrichment of viable cytokine secreting cells. Upon stimulation, cells are being labeled with a catch reagent, which on one side attach to the cells and on the other side catch the secreted cytokines. In example, interferon gamma, TNF alpha, and it can also be catching other interleukins. In the next step, the immobilized cytokines are then labeled with a fluorophore conjugated antibody named the detection reagent. This fraction can be detected in a flow cytometer or can be further enriched by a third step and using an anti-fluorophore magnetic microbeads. This is one way of enrichment. The second way is to use the manually cell sorting with either the CD154 microbeads or the CD137. The CD154 is transiently upregulated upon activated T cell of the CD4 population, whereas the CD137, which also called the 41BB, is a suitable activation marker for CD8 T cell population and early activation marker for Treg. Both workflows are very established and highly cited. The secretion assay is also being used for several years in the clinics and is approved for the virus-specific T cell enrichment application, called also the VST. Then what is the importance of the T cell as antivirus responders, taking it from research to clinic. Until this point, we have reviewed the T cell section, which were the analysis and the isolation step. And from now, we're going to see how this application in the research field come into action for the clinics. In the clinic, it's important to monitor the efficacy of newly vaccines, to check the prognosis of the disease progress and adjusting the treatment option for SARS infections, analyzing the immune population in epidemiological studies, for T cells as the target point for vaccine developments, testing and evaluating long-term production by virus-specific memory T cells. And in the next slide, I'm going to show you the clinical workflow. Here, PBMCs are isolated from a patient. Antigen-specific stimulation is performed in vitro. Then there is an addition of antibodies for the phenotyping and analyzing on a flow cytometer, or for the sorting and enrichment of those antigen-specific cells. In this schematic workflow, the MAX1 titer cell sorter is demonstrated for its low pressure and gentle cell sorting, and also for being approved for GMP compliance cell sorting. In this last slide of the antigen-specific T cell section application in the clinic, I wish to present you with results from a published paper on a successful manufacturing of clinical grade SARS-CoV-2 specific T cells for adoptive cell therapy. This clinical workflow has been performed on the automated Clinimax Prodigy using the interferon gamma cytokine capture system, also known as the CCS. This figure presents the T cell immunophenotyping before and after CCS enrichment. T cells from donor 1 were assessed for interferon gamma, and the memory cell phenotype also present on the right. Both were analyzed by flow cytometer before the cell selection, which was the original fraction, post-enrichment in the positive fraction, and also in the negative fraction. Separate analyses were performed for CD3 positive cells, CD4 positive, and CD8. Panel was gated on the interferon gamma subset, as shown in both panels. Here, we have arrived to the B cell and antibody section. In the past year, publications have been emerged presenting the effect of COVID-19 virus and the newly mRNA vaccines on memory B cells, neutralization antibodies, and the entire B cell response. Researchers have been tested samples from patients, convalescent, and from mRNA vaccinated volunteers. The results point out that few weeks after the second vaccine injection, high titer levels of IgM and IgG are detected against the spike protein and the receptor binding domain. Moreover, the plasma neutralization activity and the relative number of the spike or RBD specific memory B cells were equivalent comparing to the individuals who recovered from the natural infection. In order to continue tracking and monitoring the efficacy of newly manufactured monoclonal antibody in the clinics, and to further investigate the potential clinic outcome of the mRNA vaccines against the newly arising variants, more research needs to be done. let's dive into the potential application of SARS-CoV-2 antigens in terms of antibodies and B cell. And here also, it can be divided into three main workflows. First workflow, demonstrated on the left side of the slide, is the SARS-CoV-2 specific antibodies. It's for the detection of those antibodies in samples from patients, convalescents, and vaccinated individuals. In the second workflow, there is the detection and phenotyping of the SARS-CoV-2 specific B cells in PBMC samples. And the third one on the right is the enrichment of those specific B cells for further culturing and other downstream applications. The basis for all three workflows mentioned are the SARS-CoV-2 recombinant proteins. On the left side, in the schematic overview of the SARS-CoV-2 antigens, this overview illustrates major structural SARS-CoV-2 proteins, as you are all familiar with, the S, which is the spike, the N for the nucleocapsid, the E for the envelope, and the M from the membrane. For the right side on the slide is the list of the available different formats of those recombinant proteins. These proteins are biotinylated for the use of the antigen-specific B cells labeling in the workflow I will present to you on the next slide. Starting with the antibody detection workflow. This is a bead-based immunoassay. The first step is the coupling of the biotinylated recombinant proteins to stratavidin-coated beads. Then, the coupled beads are incubated with plasmapheresis samples, where the last step is the addition of anti-Ig-kappa PE for the detection of the multiple antibody isotypes in flow analysis. Here is an example of an analysis of different plasma dilutions. Illustrates the highly sensitive detection of patient-derived anti-SARS-CoV-2 antibodies enabled by the bead assay. In this example, all 4 biotinylated recombinant proteins were coupled to stratavidin microbeads. The ACE2 serves as the negative control. After incubation with plasmapheresis samples of COVID-19 patient or with a healthy donor, captured antibodies were detected with anti-Ig-kappa PE, and the flow cytometry analysis was performed. Now, we are going to talk about the next 2 workflows, which are the detection and phenotyping, and the last one is the enrichment. In order to perform the detection and enrichment, the basic and first step is the tetramerization working protocol of the SARS-CoV-2 recombinant proteins. The creation of this complex is the first step for the isolation and detection of the antigen-specific B cells. The fluorophosphatavidin conjugates is incubated with 4 molecules of a recombinant SARS-CoV-2 protein of choice. Afterwards, these tetramers are introduced to the isolated B cells, and after incubation, they can be analyzed. After performing the tetramerization step, untouched isolated B cells are labeled with a tetramer complex and can either be detected for the antigen-specific B cells by a flow cytometer, or to be sorted by a cell sorter, or even manual sorting for further culturing. It is important to isolate the PBMCs as untouched cells. The isolation can be performed with either pan-B cell isolation or with acidic human release beads from Miltenyi Biotech. Here is an example of a detection and phenotyping of spike-specific B cells. In the table, there is a recommended flow antibody panel, which you can see here in the middle, in the upper row. The results presents a flow analysis using antigen-specific B cell isolated using the Realis CD19 microbeads kit and analyzed with the recombinant engineered antibodies from Miltenyi. The results show those double-labeled antigen-specific B cells. Whereas on the top, there is a graph that sums the percentage of antigen-specific B cells from COVID-19 convalescent samples comparing to healthy donor samples. As you can see in the plots on the bottom, the spike tetramers need to be double-labeled. It needs to be a PE and also be detected with the PE size 7 because this is a very rare population. Another example for the use of the protein tetramer is the isolation of the labeled antigen-specific B cells via a cell sorter. Prior to the sorting, the enriched B cells are incubated with the fluorescent protein tetramers and the phenotyping antibody panel. A recommended panel is exhibited in the table on the left. Those results were obtained with the MaxQuant Taito sterile closed system cell sorter. In the flow analysis, you can see the gating strategy. After gating on the population, live cells are gated, and then 2 gates can be drawn for spike positive cells, which is the upper gate, gate A, whereas the lower gate is the rest of the B cells, sort gate B. These sorting results were obtained on the MaxQuant Taito. At the end of the sorting, both fractions are being analyzed. The results are presented below. The positive fraction of the antigen spike specific B cells and also the negative fraction of a healthy control. One of the downstream applications is the spike-specific B cells, culture and IgG production. Prior to culturing, B cells need to be enriched via sorter. Afterwards, the positive fraction can be expanded in culture for the production of antigen-specific antibodies. For the B cells expansion, there is a unique kit containing all the necessary supplements, growth factors, and cytokines. All the products that were presented in the B cell section will soon be available as kits. One for flow analysis and sorting, and the second kit for magnetic enrichment. Are there only T and B cells in virus research? Of course not. You can perform NK isolation and phenotypic analysis with one of the 3 recommended panels below presented in the table. You can also isolate plasma cell from virus-infected individual and analyze them. This is the end of this webinar. I wish to add that we are performing CRO flow services, and we will be happy to guide you and to tailor the most suitable workflow for your research or clinical system. If you are interested in the COVID-19, in other COVID-19 products, I invite you to see our previous webinar on the COVID-19 toolbox available in Almog webpage. If you have any questions, please feel free to write them to us via mail or to call us. Thank you.